Today we are going to learn about the next practical, bacterial transformation. Before going to the bacterial transformation, why we need to do the transformation? To make any genetic manipulations to the organism to achieve the targeted protein or targeted recombinant DNA technology, we need to do the bacterial transformation where the insulin deficiency in the human beings because of the diabetic, where we require the insulin. Unfortunately, the people's not able to produce the insulin. We are getting it in the form of the injection from external source. So here, how we can incorporate the gene of interest into a bacteria, because bacterial membrane not easily penetrate the targeted DNA. So to make the bacterial membrane to be penetrate, we need to give the permeability by giving the calcium chloride mediated treatment. Then the cell membrane will be allow the targeted DNA into the bacteria. Bacterial transformation practical is a 3 days practical where the day 1 we collected the E. coli from the kit. It was placed on the inoculated into the LB agar plates and kept it for 37 degrees Celsius for the overnight. Then the second day we already performed with inoculated a single colony on the nutrient agar, the LB agar plates. Now we are in the third day where now I am preparing the LB broth for the next step, where I need to be inoculate a loopful of a colony from the collected already from the day 2. Now I am going to prepare the LB broth, where I need to take the 1.38 gram of LB agar. Now I am going to add it 0.5 gram now. Still I need to add some more. Then I got 1.5 gram. Now carefully we need to transfer this powder into the conical flask. The conical flask which I am going to be do later on for the autoclaving. Once we did, we need to be mix it very well. Gently we need to be mixing. We need to be mix it well. Then we need to add the balance water of 55 ml. Total now the 55 ml of the luria broth is ready. Further we move on to the autoclaving. Now we are going to the autoclaving process. Now we are going for the autoclaving process. Now this 55 ml of the luria broth already prepared is going for the autoclaving. This is the setup for the autoclaving, where it will maintain the 121 degrees of the temperature and 15 lb pressure will be maintained, which is a sterilization process to be performed for autoclaving process. It will take around 1 hour plus time to do the autoclaving process. Then we will come back to take our samples later. After the autoclaving process is finished, now you are going to take out the product from now we are moving to the biosafety cabinet to pour it on the petri plates. Now the media which is prepared in the media preparation room, now we brought into the area biosafety cabinet. Here we are working in this biosafety cabinet. We need to take the petri plate, gently hold the petri plate in this hand like this. Then we are going to pour the media into the petri plate, which may be required around the 15 ml for the each plate. Okay. Once we did, we need to leave the plate it is to be get solidified. It will take around 15 to 20 minutes of time to get into solidification. Then we will leave the plate in this biosafety cabinet for the 15 to 20 minutes. In continuation to the day 3 for transformation, here already the yesterday's overnight grown culture is here. Now I put it for the pre-chill. Now I am going to transfer into a centrifuge tube, which is a microfuse tube. The overnight culture, which is after getting the OD value of 0.6. Now I am going to transfer this culture into total of 2 ml transferred. Once we transfer to the tube, we need to be cooled down at the 4 degrees temperature for the 10 minutes. Now we are going to leave this for the 10 minutes in this ice box, or we can keep it at the area where we can maintain the 4 degrees Celsius temperature. So now we are going to leave it for 10 minutes of time. After the 10 minutes of incubation time is finished, now we are going to take the tubes for the centrifugation, which is at 5000 rpm for the 10 minutes of time. Now we are moving to the Remy centrifuger to do the centrifugation. Once we set everything, now we need to give start. Then the centrifugation started. It will be stopped by 5 minutes of time. So after the 5 minutes, we can take out our sample out. After the centrifugation finish, the first step, we are going to take out our sample. After centrifugation, we need to completely discard the media, only the pellet need to be left over. So we need to carefully remove the media, only we need to allow the pellet which is deposited at the bottom. So gently we need to do this process, no need to disturb the pellet. The pellet is deposited at the down bottom. Now to this pellet, now we are going to suspend this pellet at 0.1 molar calcium chloride solution, where we are preparing these competent cells. So to these competent cells, already the kit provided with 0.1 molar calcium chloride solution. Now I am going to add this chilled 0.1 molar calcium chloride solution. this total 2 ml of calcium chloride solution is added. Then now we need to incubate this pellet on ice for the 30 minutes of time. Now I left this for the 30 minutes of time for the incubation. After the 30 minutes of time, now I am going to take the sample to go for the centrifugation again at 5000 rpm for the 10 minutes at 4 degrees Celsius. Once we done, go for centrifugation. For the 5 minutes, it need to be go with 5000 rpm. After the 5000 rpm finish, now after centrifugation, we need to discard the calcium chloride solution, which is just now after the centrifugation where the competent cells are getting ready. Now we are removing the supernatant, which is the complete calcium chloride solution. We are going to take it out. After this, now we need to resuspend the pellet with again the same 1 molar calcium chloride solution. We need to add 2 ml of the calcium chloride solution. Then we are going to be adding, then we can be used for the cell suspension. Now I added the 2 ml of pre-chilled sterile 1 molar 0.1 molar calcium chloride solution. Now the cell suspension contains the competent cells. Now the competent cells are ready. This is the competent cell preparation. What we did, just I summarize. The first thing, from the overnight culture, the E. coli culture provided by the kit, we transferred to the luria broth. From that luria broth, we did the fresh culture. Within the 3 to 4 hours of time, we reached to the 0.6 OD. Then now from that we take the sample and we first did the centrifugation to get the pellet of the bacteria. Then the medium is completely discarded. Later we added ice cold 0.1 molar calcium chloride solution. Then we went for the centrifugation at 5000 rpm for the 10 minutes. Later we discarded the total calcium chloride solution. Then later we added again the calcium chloride, which is ice chilled in this ice box. Now the competent cells are ready. Further we move on to the transformation of the cells. We will move to a biosafety cabinet for the further experiment. Once the competent cells are ready, now we are going to perform the transformation. The competent cells, from that prepared competent cells, now I am going to take 2 tubes, which are the normal centrifuge tubes, where I am going to take 2. One I am going to mark it as a control, another one I am going to mark it as a transform. One is a control, another one is a transform. To this, we are going to add 200 ml of the competent cells. I am going to take the 200 ml of competent cells. From here I can fix the 200 ml. Now I am going to transfer the competent cells which are prepared just now. From there I am going to take 200 microliter into the two tubes. One tube I marked it as control, another tube I marked it as, where I marked them, one as a transformed, another one as control. Now incubate these tubes on ice for 30 minutes of time, where I brought the ice box here to place them on the ice, or you maybe use the ice packs to be keep them for the 30 minutes on ice for incubation. Now I left it for the 30 minutes of incubation. Now the 30 minutes finished, we are move on to place these competent cells which are kept on the ice. I am going to heat them. Before that, I need to add the 2 microliter of plasmid DNA to the transformed cell, the transformed which I mentioned just now. I need to be transferred 2 microliter of the plasmid DNA. To the one tube which I mentioned it as a transform, I am going to add 2 microliters of the plasmid DNA, and I left it for incubation for the 30 minutes. Now the 30 minutes time over for this incubation. Now we are moving to make the preheated water bath setup for the 2 minutes to give the heat shock. Where I placed it for the 2 minutes of incubation. Incubation is finished. After the incubation finished, now rapidly transfer these tubes onto the ice bath. Now I kept it for ice cold bath for the 5 minutes of time. Again I use the ice box for the cooling. After this, I am going to add 800 ml of luria broth. 800 ml of luria broth I am going to be add to the both the tubes. For the transformed cell and also for the control, just I am adding 800 ml of luria broth. Now we need to incubate these tubes for 1 hour at the 37 degrees Celsius. Either we can leave them at the room temperature, or we may be keep it at our incubator. Just where I am going to be keep them in the incubator.
1, 2, 3.
After incubation for 1 hour for this control and transform, now I am going to transfer these control E. coli cells and transformed E. coli cells to the Petri plates, which are the LB agar plates containing ampicillin, Xgal and IPTG. To this, one is marked it as a control, another one marked it as a transform. From the control to the control, I am going to add 200 microliter of the sample to the plate. Then I will transfer the 200 microliter. I can fix it as now the 200 microliter of sample with me, where I am going to place it at the center of the plate. Then by using L rod either in the clockwise or anti-clockwise direction, but follow the only one direction. It's always suggested to use in the clockwise direction. Make sure the L rod should be touch the surface of the agar. Don't press it harder. Gently need to make spreading, where you place at the center and turn around. Now the bacterial colony, which is transformed with plasmid DNA, is transferred to the plate. Mention it as a transform. Then now I am going to take the control cells. These control cells, I am going to take 200 microliter of the control. The control organism, which is not added with any plasmid DNA. Again, place it at the center of the plate. Then by using a new L rod, we are going to be spread it in again in the clockwise direction. Once the spreading is done, now we are going to place these Petri plates are ready now. We are going to incubate it for the overnight at 37 degrees Celsius in our incubator. Now I am going to place it in the incubator, and we will see the results tomorrow.